This is Biosphere, a Royal Society of Biology podcast that covers the broad field of the life sciences by interviewing bioscience researchers and discussing interesting biological discoveries and science policy. Welcome back to Biosphere. I'm your host Freya, and this will be our very last episode of this first series. I know it's very sad, but we're ending this series with a bang, as this is our Thinking Outside the Box episode. We will be looking at weird, quirky research, the unexpected, and I'm joined today by Claire to help me explore this topic.
Thanks for having me.
Claire is on the same team as me, the Comms and Parliamentary Affairs team, and if I do say so myself, it is the best team at RSB, wouldn't you agree?
Yeah, without us, no one would know what the RSB are up to.
Exactly. So for this week, I found a very interesting article about squash bugs.
Yeah?
And their poo.
Wow. Okay. Do I want to know more?
So probably not. No, you do. So squash bugs, a common and difficult to control agricultural pest, need healthy bacteria in their gut to grow and stay alive. Now, normally most animals will have their gut biome passed down to them through vertical transmission. So in other words, it's passed down directly from your parents. This is also similar to when humans get their mitochondria, the powerhouse of the cell, from the maternal lineage. Just shows how powerful women are. Am I right?
That's a terrible joke.
You laughed. So
that's vertical transmission, right? But there must be horizontal.
yeah, so horizontal transmission is when you gain something from a different individual. A classic example is getting the flu by being infected by a friend when they pass on a virus to you. This is obviously an example that is affecting you in a negative way and you're not intentionally trying to get the flu, but this is the general gist. With squash bugs, they do not acquire any gut biome bacteria from their parents when they are first born, so there is no vertical transmission. Instead, they, and this is a direct quote from one of the researchers, seek out feces and slurp it up.
That's gross.
Yep. Yeah, it is. It is very grim. But what the researchers found is that these squash bug nymphs, or babies, only eat the fecal matter of adult squash bugs. They were searching out these necessary bacterial symbionts in the environment because ultimately they would die otherwise. And that is why this research is so important. By understanding this early stage of development in squash bugs, we have identified a vulnerability. The discovery of squash bug species-specific fecal preference might be helpful in developing pest control against them. If we can target them at this vulnerable stage in the life cycle, that's a really efficient way to target a specific agricultural pest and nothing else in the ecosystem, which is something very important that we discussed in our plant health episode earlier on this series.
Who knew that poo could be the answer?
I know, very weird research, but it does also beg the question, what came first in terms of the bacteria or the squash bug? Because if the parents don't have it, it's like a chicken and an egg situation. So at some point, was it not species specific and they actually found other species bacteria from fecal matter? But it seems like it's a step in the right direction to find alternatives to pesticides.
Indeed. Well, solutions to many of our most pressing problems arise from kind of this out-of-the-box thinking. Like, would you predict that fungi could be the answer to our need for sustainable fireproofing?
No, I didn't. Tell me more.
You're probably aware of the tragedy that occurred at Grenfell Tower due to the flammable cladding used in the tower's construction. Researchers have been searching for a suitable material for sustainable fire-retardant cladding, and the answer may be fungi.
That's really cool.
Yeah, so basically, a novel method of growing mycelium in sheets has been found, and it produces a paper-thin sheet that actually forms a thermal protective char layer when it's exposed to fire or radiant heat. So this research could eventually lead to improved and eco-friendly cladding for buildings. So currently, composite cladding panels are used, and these are effective at repelling flames, but they usually contain plastics, which actually produce toxic fumes and heavy smoke when they burn, and they pose health and environmental concerns. Bioderived mycelium, on the other hand, actually only produces water and carbon dioxide.
This sounds too good to be true. There must be a drawback, right?
So, fungi is very slow to grow at scale. So, researchers are actually connecting with the mushroom industry to discuss using their fungal incorporated waste products. So, this would remove the need to produce new farms and help to meet fire safety needs in a sustainable way.
So what's the next step in the research?
So the researchers are now looking to create fungal mats reinforced by engineering fibers to delay ignition, reduce the flaming intensity, and improve fire safety ranking.
Well, that's really fascinating, and it's great to hear about such interesting science that will ultimately keep our community safe. Speaking about innovative research, which I mean is the theme for this episode, I think it's time to bring on our special guest. Today we have one of our very own RSB chartered biologists joining us, Steve Trim, who is the founder and chief scientific officer for VenomTech, the UK's only commercial venom lab serving the globe with venom-based peptides for drug discovery, pest control, and cosmetic applications. Thank you so much for joining us today. Is it right that you offer a library of around 20,000 peptides derived from venoms?
Yeah, so thank you for the invitation. It's really great to be here. And yeah, we've got approximately 20,000. I say approximately because we've never counted them all, because the venoms are complex mixtures, and it's only when we investigate a particular venom species that we separate it out into its component parts and find how many are there.
Wow, that's mad. So just for the purposes of our audience, when you refer to peptides, what do you specifically mean?
What I'm talking about when I'm talking about peptides, I'm talking about small proteins, basically, before they become enzymes in the size scale.
Okay, so as I just mentioned, VenomTech is the UK's only commercial venom lab, but you spent a decade working as a molecular biologist and drug discovery at Pfizer researching pain therapeutics. So there are so many biological compounds in the natural world. Why focus on venom and start a company to research this?
That's a very good question, and it came from a very unique set of circumstances. So yeah, as you mentioned, I was at Pfizer for 10 years, and about 8 of those were in pain and neuroscience. And the challenge there at that time, and it's still a challenge now, is getting good compounds that are selective, so they do good pain relief but have low side effect burdens. And something obviously patients are very, very keen on. Because small molecules are small, so typical drug-like molecules, they often have a challenge binding one pain ion channel and not a cardiac or brain ion channel. And that's where the origin of the problem comes from.
So, for context, the small molecules that you were referring to are typically used in drugs that affect molecular pathways by targeting important proteins. And here we're talking about ion channels. So, ion channels are proteins that are prominent components of the nervous system. And in this case, ion channels are critical all along the process from detection to final interpretation of painful sensations.
Yeah, so peptides are larger, and therefore because they interact with more of the channel, they can improve the selectivity. So that's what led me into venoms, is actually trying to get better pain therapeutics. But what then led to VenomTech was another couple of circumstances. One was that I was the safety officer for toxins, charged with the sort of safe working practices for that as part of the pain team. And the other was I understood the animal biology. So I've been keeping non-dangerous spiders and snakes as pets, and I still do now. And so when Pfizer decided that myself and many of my colleagues needed something new to do, I took those 3 skills and set up VenomTech to supply venoms for drug discovery. So we actually went out and collected a load of venomous animals, mostly from captive bred stocks, to actually then build that process. But it's interesting you're talking about pain, because a lot of people will be familiar with how venoms are acting, but it's actually when you take them apart into their component parts is the real thing. And some things do cause pain, and some things are interestingly analgesic, but some of that's serendipitous. So most venoms didn't evolve to have an impact on humans. We're quite late in the evolutionary sphere. So the targets that evolved in the predators and prey of the venomous animals also then owe a lineage to our evolution. So we have origins of our proteins in our body that come from our ancestral species, which then the venoms are acting on. So there's a bit of serendipity in there as well, which makes it for a really interesting mixture. So I came into venoms purely to focus on pain and ion channels. And then in this world, I found them applicable to a much wider range of biological applications. And that's spun it out into sort of 3 core markets, really.
It's interesting because most people would associate venom with negative associations, with pain, being bitten. But here we're discussing it within a drug discovery context. What kinds of drugs have been formed based on venom peptides?
Yeah, it's a challenge for us as a company that most people look at that negative association, but all pharmaceutical drugs are toxic at some level. It really depends on the dose partly, but also the context of pain. Pain is a strong learning tool. So animals that are defending themselves will often inflict pain because when the predators are feeling pain, that makes them avoid that interaction. And some of those pain-causing toxins can be useful as tools if we want to study how ion channels work. But also, there's a lot more going on. So some of the first drugs to come from venoms are actually working in the same way that the venom does on its prey. So captopril, which is the ACE inhibitors for hypertension, owes its entire discovery to snake venom.
And ACE inhibitors prevent an enzyme in the body from producing angiotensin II, which is a substance that narrows blood vessels, right?
Yeah, so peptides from the fer-de-lance snakes, the Bothrops genus, actually cause a catastrophic drop in blood pressure, and that's part of their predator activity. And through studying this, scientists sort of brought some venom over to London to actually understand how this mechanism worked. And through understanding that, we understood about blood pressure regulation. And then captopril is actually a small molecule that mimics that process and actually can then be taken as a tablet, as a drug. But you've also got Priel. So this is a cone snail peptide. And cone snails are fascinating. They are little squidgy marine snails that have evolved to feed on bony fish. And the only way a soft squidgy snail can feed on a bony fish is if it kills it very, very quickly. So it's evolved very potent ion channel blocking peptides that just immobilize the fish, and therefore the snail can then slowly engulf it. But this class of ion channels are really useful in our central nervous system pain processing. And so the ziconotide has to be injected into the central nervous system, because if it's in its systemic circulation, it does cause cardiac problems, which ultimately is part of its original evolution. But that really potent ion channel blocker has given us a really powerful pain medication that has much fewer side effects, particularly when you're comparing to morphine, but has come from the animal's own action on how it kills its prey. And understanding that biology is absolutely fascinating. It's one of the things that really excites me with what we do. And sort of drug discovery starts to unpick new mechanisms of how venoms are signaling. And then one of the other things we did was a marketing project a few years ago. There are venoms from tarantulas that activate TRPV1. So this is the ion channel responsible for heat pain from capsaicin from chili peppers. And so when they bite mammals, that pain response will be like an injection of Tabasco sauce. And we then, with one of our partners, actually produced a venom-inspired chili sauce based on the action of that venom activating TRPV1 as an interesting marketing tool, which was quite a funny diversion and something I never thought I'd be getting involved with.
I'm going to have to try that hot sauce. So when I did biology at university, I learned a little bit about venoms. And one of the things that we learned is that they have antimicrobial properties. Is this something that you've looked into with your research?
Yeah, the antimicrobial peptide is something we do have a great interest in. Antimicrobial peptides in venoms have been known since the late 80s. And this is from classical drug discovery processes of putting venoms onto microbial cultures and observing the effects. And because they were toxic to bacteria, the dogma was that venom must be sterile. But work we did with the University of Northumbria actually details why venoms have evolved antimicrobial peptides. And that is that the venom gland is not sterile. It has a venom microbiome within it. So there are bacteria that have evolved to deal with antimicrobial peptides. And so the venomous animals are being forced to produce antimicrobial peptides to control an infiltrating microbiome, basically. And we focused in on Enterococcus faecalis as a bacterial species because they're quite well characterized both in human disease and in other systems. The ones from the venom gland were many times more tolerant to venom than the clinical isolates. So the cobra venom was lethal to the clinical isolates, where the isolates from the venom microbiome gland could actually survive in the venom. So we were able to put a piece of the puzzle together of why venoms have evolved antimicrobial peptides. In this, we found a lot of microbial gene sequences that are not in the databases. And these antimicrobial peptides can be very useful for antibiotics for a lot of systemic infections in humans. But if you're looking at a potential infection from a snake bite, then you're going to need something different because the bacteria have evolved to tolerate the snake venom proteins.
Is this almost like a secondary infection after the actual bite?
Yeah, so people were seeing snakebite infections as a secondary infection because they believe the venom to be sterile. Whereas what we think is happening in some cases, at least, is that some of these infections are actually primary from the injection of the venom, whereas secondary infections would be less likely to be tolerant to the venom peptides.
Okay, that makes sense.
And so it's a whole branch of biology that we were able to unpick, and there's a consortium called IVAMP now looking into how wide this is. So we found it in snakes, spiders and scorpions, but microbiomes have been found in venoms of snails and many other organisms as well. So it's a core part of venom biology, it turns out.
And this sounds like it must have impact on treatments of venom bites specifically.
Yeah, very much so. And one of the key things that we discovered is when you do the biochemical strain detection test strips that they often use in hospitals, that the Enterococcus faecalis actually turns up as a different species because of these different genetic elements within the bacteria that help to evolve tolerance to the venom. So you actually need to do gene sequencing to find out which species are invading from the venom. So yeah, it is a key thing. It's not the top concern from a venomous snake bite in particular, but for the long-term health of the patient and recovery, it needs to be considered.
That's really interesting. And I wanted to touch on the fact that your team demonstrated tarantulas respond to environment enrichment in a similar way to mammals, and you even won 2 awards for this work. I just wanted to get an idea what environment enrichment is for our listeners and what are the implications for animal welfare.
Yeah, this is some very early work we did. And one of the key things of having an animal lab is making sure that the animals are in the best possible health to get the best possible science. And what we looked at is that a lot of our suppliers were just keeping spiders in small plastic boxes with just a bit of substrate in the bottom. And we sort of questioned whether, is this all they need, or is there more going on? Environmental enrichment is actually how you, in captive conditions, improve the environment the animals are living in, both in terms of their physical structure, but also in terms of the mental and social health of the animals. Now, this gets really challenging when you get to invertebrates. The same things apply, that they need to be in as most natural state as possible. With rodents, they often have toys and hides, wheels and those sorts of things to keep them active and mentally stimulated. With spiders, what we found, particularly with arboreal spiders, so these are tree-dwelling species, they do need a 3-dimensional tree-like structure. So what we used was plastic plants from the florists, and animals that were in enclosures with these 3-dimensional structures behaved a lot better. And so we actually had to start off by working out what is normal behavior in spiders, because that wasn't published, wasn't known. So we found that those with the actually enriched 3D structure behaved much more as we would expect them to in the wild. They were relaxed more often when they weren't stimulated, fed better, and all these sorts of things. So, and this was quite revolutionary, but for me, it just seemed a logical thing to investigate, to make sure that we're keeping the spiders in the best possible conditions. And yeah, we got the most significant impact on animal welfare award from the Institute of Animal Technologists.
Yeah, that's fantastic. As you said, the needs of animals is really important, so much so that at RSB we advocate for the 3 Rs, which are replace, reduce and refine when using animals in research. Do you think it's possible to create and use synthetic venom?
It definitely is, because we do already. So the scientific community are very familiar with the 3 Rs. And for us, the need to use the animals is because we don't fully understand everything that's in all the venoms. And we keep just small colonies of each species to be able to have enough venom to actually fractionate it out, so separate it out into its all component parts, and then understand those peptides. And once we've got a sequence of a peptide and how it's folded, then we can make it. There is a couple of reasons why we're doing this. One is that if our clients are finding a drug or a new biopesticide, the amount of animals needed to produce that venom is just not feasible. So it would, from a practical point of view, would have to be made synthetically at that point anyway. And so actually understanding the sequence and making it synthetically early on also gives us the ability to understand the sequence. Once you've got that sequence, you can then make changes. And the wonderful world we live in at the moment is that we can find the sequence from a venom peptide, see how it's interesting, compare closely related species to understand which sequence is best, got the best pharmacology and best stability. And if we want to make changes, we just email a slightly different sequence to the suppliers and say, make this one, please. And it comes in the post a few days later. So absolutely, that we need a small amount of the species to understand the biology and find these new sequences. But then as soon as possible, we'll move to synthetic versions for the scale-up side of things.
It's just so fascinating. I mean, you mentioned biopesticides there, and we're discussing drug discovery, but of course, you also work with cosmetics and crop protection. Could you discuss some examples of that work?
Yeah, and it's really interesting because our team have come principally from drug discovery world. That was our core focus and need. But the other two markets have just come to us. Cosmetics first. So a very good friend of ours was a cosmetic chemist. And he said, oh yeah, they really love venoms in cosmetics. And I was like, you're going to have to show me how. This is not a market I understand. So the same principles apply, but it's taken quite differently. So understanding a peptide that blocks a receptor. We mentioned TRPV1 early on and pain processing. So our first cosmetic is called Sensimone P5, and that actually blocks TRPV1 to soothe irritation of sensitive skin. Whereas switching tech onto biopesticides and crop protection, this was another market that came to us, but this was customers saying, well, we know you work in drug discovery, but can you help us? We've got compounds that are getting restricted through legislation because of bioaccumulation or lack of specificity. So things like insecticides and neonicotinoids poisoning bees and other pollinators, for instance. That's what sort of drove us to look into this further, because venom peptides have some really cool properties when it comes to being used out in the environment. They are proteins, first of all, and therefore they'll be biodegraded by bacteria and very unlikely to actually go up trophic levels or be a problem in watercourses. There's a lot of protein just naturally in watercourses that's being turned over all the time. So it's very unlikely that a peptide pesticide will actually be an environmental problem. So that's significantly better than the quite persistent compounds that are out there at the moment. But also these peptides are really quite specific. What we're looking at in the biopesticide world is specificity across species, which naturally have mutations in their ion channels through their different evolutionary paths. So there's a great opportunity to actually target the core groups of species that you're interested in that are problematic for humans. If we can reduce the food that's eaten by other animals, then that ultimately makes it more for us and more efficient. And if we can then do that without poisoning pollinators or aquatic organisms or those predators that are out in the natural world, then we've got something really great.
So basically you're saying that venom peptides are the future.
Of course.
It sounds like we need to bring you back for another episode just on this, actually.
Yeah, definitely. We really appreciate you coming onto the podcast, Steve. It's been so interesting and has been a great example of thinking outside the box. We wish you every success with this research and thank you again for coming on.
Thank you both. Really enjoyed talking to you.
It's now for our last silver lining of the series.
And we have a great one for you.
Yes, a new examination of dolphin vocalizations found that mother bottlenose dolphins whistle at a higher pitch when talking to their offspring, exactly like human mothers do with theirs.
The researchers at the Sarasota Dolphin Research Program used spectrograms to study the contour and bandwidth of every call. Every dolphin has a unique whistle, and the spectrogram showed that the whistling directed at the calves had lower lows and higher highs.
We actually have a comparison here of the audio that the scientists recorded. So this is a mother dolphin call without the presence of a calf.
And this is in the presence of the calf.
That is just adorable. I'm sure you can hear the difference. I would say that's actually how I would talk to my dog. And I don't have a child, but I talk to my dog like that. So
The millennial child.
yeah, a millennial child. Well, that's it this week from me and Claire. Thank you again to Steve for speaking with us about the potential of venom peptides. And Claire, you've been a star in this final episode.
Thank you so much. I've had a blast.
And thank you to everyone who has tuned in this series. It's been so much fun creating this podcast, and I can't wait to see what interesting things we'll be talking about in the future. Until next time.